I'm Andrew Harman. I'm an associate professor here at the Westmead Health Precinct with the University of Sydney. I have a number of roles here. I'm the Applied Medical Sciences Honours Coordinator for the whole Westmead Health Precinct. But in terms of my specific research, I have two lines of research here at the Westmead Institute for Medical Research. Firstly, I investigate sexual transmission of HIV, and secondly, I look at inflammatory bowel disease. But the specific aspect of my research, which is unique to me, is that I do all of my work using discarded human tissues from operations that happen here at Westmead Hospital. Yeah, so I think modern science philosophy probably does stem back to the Greek, some of those ancient thinkers. My personal passion for science has always come. I've always, since a very young age, I've always been really bothered by things like what's the meaning of life, what's the point of it all. You know, is that do we just live and get old and die? So those really big philosophical questions for me, what is the meaning of life, for example, have always really bothered me. I think I had my existential crisis when I was about 7. And I think I felt, to me, the two obvious things that I was drawn to, which would kind of help me resolve those issues, were science, that sort of thirst for truth, and also religion. So I was very interested in both of those things. And in fact, when I went in England, when you do A-levels, I grew up in England, you have to choose 3 subjects. And the 3 subjects I wanted to choose was biology, chemistry, and religious studies. Because I was at quite a small school, religious studies and biology clashed, so I had to pick one, so I went for biology. I ended up doing physics, chemistry and biology. So yeah, so for me personally, that's where that comes from. And I think that's why in later life I became a Buddhist, because I was very inspired by the life story of the Buddha, who had a similar existential crisis, and he gave up everything and devoted his whole life to seeking truth. And I felt quite inspired by that, which is why ultimately I chose to become an ordained Buddhist as well as a scientist. So things that are currently influencing my thinking and that have probably also influenced me over my scientific career are really reading about the historical figures in science and reading about their life stories. But particularly recently I read a book called Reality is Not What It Seems, which I really enjoyed. And I feel very, very drawn to Albert Einstein. And I think one of the things that I love about people like Albert Einstein is that they were very, very bothered by theories that contradicted each other. So for Albert Einstein, he was very bothered by the fact that Newtonian mechanics, which although were very sound and described reality from a certain perspective, didn't agree with electromagnetism. So he had to come up with the theory of general relativity to actually kind of, which kind of provided a network where both of those things could be true. And I think a lot of the really big scientific thinkers have been really bothered by that. And that's something that I find inspires me, is that now in science, we have lots of different people around the world doing different research, and a lot of them with very high integrity and great research that I really respect, but who come up with completely different answers to questions. So I find that really sort of bothers me, but also inspires me to work out, well, what's going on? So my own main field of research was sexual transmission of HIV. I've been looking at a particular cell type called a dendritic cell for the last 15 years of my life. And we find that cell transmits HIV. It's a key cell that binds to the virus and transmits it. There's another research group in the Netherlands who publish in very, very high-ranking journals, and their conclusion is that these cells inhibit transmission of HIV. So to me, it's like, well, hang on, what's going on there? Because I respect the integrity of that group, but I can't agree with what they've done. So in a recent research paper that we've just published in Nature Communications, we really went back and examined the methodology by which we both do our experiments, and I found that there were differences. And by delving into those differences, I was able to discover that actually the cell type we were working on was actually 2 cell types, and that one of them was being misidentified. So we've actually just published methods to actually delineate those 2 cell types. And actually, indeed, one of them transmits HIV, and one of them probably does block the transmission. So it was just that we were both doing great work, but it actually required that sort of in-depth. And you have to be really bothered by it to find that out. Most people would just sort of go, oh, whatever, it's just contradiction. But it's that. And I do find those really great scientific thinkers. It seems to be what motivated them. I know we talk about science in that way, like hypothesis-driven research, but I find that's not the reality. I mean, yes, we do have hypotheses, but in terms of initial hypothesis, I don't think I could go back and define the moment where I had an initial hypothesis. I find that basically just the research just evolved. So you start looking at one thing, and usually 9 times out of 10, it doesn't go the way you thought, and just new questions arise. It develops from an initial observation that perhaps was unexpected. Yeah. So you then think, well, I wasn't expecting that. Well, what are the possible reasons why that might happen? So you might hypothesize that it happened because of this reason or that reason, and then you, of course, you have to then design the experiment to really look at to say, well, is that true or is it untrue? Well, generally it's in collaboration with others. So generally it'd be something that I would discuss with my mentors, with my research team. But really it's assessing, reading the literature, working out what equipment do we have available, what tools have we got to look at this, and then really trying to pick the right tools, the right people, to delve as deeply as possible into the question. But I think keeping an open mind is really important. Ideally you have an attitude of, how can I prove myself wrong? Because if you're trying to prove yourself right all the time, you generally get much more biased. So being open to the fact that what you think is wrong, because most of the time you are wrong. That's the reality. I think it is just something that just happens. I mean, a lot of it. I think for me, one of the defining moments about when I quite specifically changed direction was when I became an independent researcher. So when you have a scientific career, for a long time you're doing your honours degree, then you're doing your PhD, and then you're doing a postdoc, but you're always being mentored by someone. So really it's somebody else's ideas that are driving it. There's then that really crucial moment in your career where you want to start your own research group. And that's very difficult, because you have to be driven by everything you've done in the past, but there are other people's ideas. So it's an awkward moment when that step into independence, because you need the support of your mentor, but you also can't just nick all of their ideas. So for me, that was when I moved into the human tissue work. I'd become an expert in HIV and dendritic cells, but that was all Tony Cunningham's ideas, not mine. So I thought, what can I do that's different? Well, actually, I can start looking at these things in real human tissues, because I work at a hospital. So I think one of the things that drives you is that need to be original, and to have original ideas, and to be competitive, in your funding. The data we produce is all derived from human tissue. Human tissues are problematic to work with, because you get incredible donor-donor variability. So when you're working with cell lines or animal models, everything's very controlled. So if let's say you're using a mouse model, it's the same strain of mouse, it's eaten the same food, it was born on the same day, and you can really, really control things, which really is great for statistical power, because you can really, really know that it really was administering that drug or doing that thing which has given that outcome. The problem with working with human tissue, so for example, we get a lot of tissue from cancer patients, for example, where we get the healthy tissue that's removed as well as the cancer, and that's our healthy control to compare to, say, our diseased tissue, so disease that's affected by inflammatory bowel disease. The problem with that is that we might have one piece of tissue from a 9-year-old, another piece of tissue from a 90-year-old. One may have had radiotherapy, one may have had chemotherapy, one may have had a different type of chemotherapy. So in terms of statistical power, that's problematic because we get a lot of variation, and you have to be really sure then that what you're seeing is a result of, say, the disease and not a result of the treatment. So really the biggest problem for us is getting enough donors to really make sure that we are seeing an effect. So to get statistical power, we need lots of donors, and also knowing when to exclude data and when not to exclude data, because sometimes you do get clear outliers and you have to exclude that. And you have to be very careful that you're not doing that on the basis of wanting to get a result, that there is a clear clinical reason to remove that data. So we might find, for example, that people who have radiotherapy consistently don't have that cell type. And then we have to then obviously go and exclude any patient that has radiotherapy from our data. So we don't do it by signal analysis, but what we do is we would look for, so I'll give you an example. So a lot of the work we're doing with inflammatory bowel disease, we're looking at inflamed tissue, okay? So we need to make sure the control tissue is uninflamed. Now there are certain markers of inflammation. So rather than a statistical tool, we would take the supposedly uninflamed tissue and ask, does it have any markers of inflammation? And often it does, in which case we have to exclude those based on those markers. Some of the things we do with those big data sets is what's called hierarchical clustering. So you get a whole group of cells that you've sequenced, and then what you need to do is you need to look at how similar are they in terms of the genes they express. And that's one of the great things about modern science, is that there's this new, I don't know if you've heard of it, there's this new Cell Atlas project. So you probably remember about 20 years ago there was the Human Genome Project where they wanted to sequence the entire human genome. Well, we can do that in about 10 minutes now per cell. What they now want to do is to define every single cell in the human body using these sequencing, and that's what's coming now. And one of the ways you do that is you look at the specific genes that each cell expresses. So a dendritic cell will have a whole load of genes which they express that are specific to dendritic cell functions. A macrophage will have macrophage genes, a lymphocyte will have lymphocyte genes, and a neutrophil will have neutrophil genes. And what that's meant is that recently lots of cells, for example, that we've been calling dendritic cells, now that we've individually sequenced them, we actually now understand they're not dendritic cells, they're macrophages, which is really change the way we can think about the immune system. So that's something that we did with this brand new cell type. We did the RNA sequencing, and then from looking at the gene signature, it was quite clear it was a type of dendritic cell, which was an important observation. So that's exactly how we were able to decipher that there were two different cell types, that when we isolated those cells and sequenced them, we could clearly see two gene signatures. And by looking at those two gene signatures, we were then able to say, okay, well this gene signature, this particular set of cells in the population expresses the protein CD11C we found. So by including CD11C in our cell definition panel, suddenly it was really obvious that one population expressed CD11C and one didn't. And then it was really obvious there were two cell subsets, and then we saw that they were completely different. So I think there's two really unique things about the work we do. As I said earlier, we use human tissues. So one of the difficulties in scientific research is making that all-important jump into human work. It's the hardest thing to do, because ultimately animals aren't humans and cell lines aren't humans. You have to do things in humans. So I think our access to human tissue really makes an enormous effect. So for example, one of the things that we've recently found is that the cell types in genital skin, for example, are very different to the cell types in skin on the rest of your body. And that's really, really important to do, because you have to know what cell types are there in order to understand how the disease progresses. So I think our access to human tissue and the methods that we are publishing to work with these tissues is really, really valuable. I think it's an important way of progressing research into the human as a first step, and also to some extent is more relevant than animal models in some ways. A second area that I think where our research is really groundbreaking is our work with viruses. So viruses are very, very difficult to research because they're tiny and they're very, very difficult to visualize. In fact, traditional imaging can't see a virus unless you use an electron microscope. You can't see them, and even when you're looking at viruses, let's say you're looking at an infected cell in somebody, you have to wait for the virus to have replicated until there's probably thousands and thousands of individual virion particles before any of the current methodologies can detect that virus. And what that is particularly problematic for is for investigating transmission, because when a virus is transmitted, whether it be via needle stick injury or sexually transmitted like HIV or herpes, the initial infection event is a single virus particle infecting a cell, and no one's been able to actually see that happen because of the need to wait for 3 or 4 days for the infection to get going. And by the time 3 or 4 days have passed, the virus has spread all around different cell types. So one of the things that my lab has done has been to use this brand new technology where we can actually detect single virions of HIV using a very, very specific probe detection system, and we can detect those virions within 30 minutes. And that has allowed us to actually witness the event that occurs when transmission occurs. And that's why this paper recently got into Nature Communications, because we've been the first people to be able to actually see single virions of HIV actually interacting with their initial cell types in the actual tissues where transmission occurs, in the inner foreskin, the vagina, the anus, etc. As long as you've got the virus genome sequence, which all viruses have been sequenced, you can design probes to that virus, and we've already adapted it for herpes viruses. In another project we've got, because I don't know whether you know, but if you're infected with herpes simplex virus, your chances of acquiring HIV are 4 to 5 times higher. So we've actually designed probes now where we can detect herpes simplex virus and HIV in the same cell type. So we label one set of probes red and one set of probes green, and we can see green dots and red dots, and we can really understand which cells are taking up the green dots, which ones are taking up the red dots, which ones are taking up both dots, and does pre-exposure with one virus enhance uptake of the other, and vice versa, all those kinds of things. We do sometimes, so as I say, one of the things we've done with these tissues is not only identify this new cell type that no one's seen before, which people were confusing with the previous cell type. So we've identified that cell, and we've also visualized it taking up single virions of HIV. But once we discovered that cell, we had to then ask the question, well, what is it? And that did involve sequencing its genome, and then also sequencing the genome of all other cells that might be similar to it, which did lead to huge data sets of basically RNA sequencing data, gigabyte files. And for us, we then have to get, because unfortunately I didn't do maths in my day, one of the things I'd say to any new scientist, any new biologist, is that maths is incredibly important, and really take maths seriously. I didn't, and I'm now suffering because I'm not really able to interpret half the data that I rely on my PhD students now to do these really complex bioinformatic approaches to model huge data sets. So the way we do that is primarily through peer review. So you send your manuscript off to the highest ranking journal that you think will take your research. Generally you aim a little bit higher than you think you'll get in, but sometimes you're lucky, sometimes you're not. But it goes through a very rigorous process of peer review. So first of all, the editor has to like it. There's usually, with the top ranking journals like Nature, Science, Cell, Journal of Experimental Medicine, those kind of journals, goes through a round of editorial review first, and if the editor likes it, which is probably about 5% of them, then they will send it to peer review. It then goes to 3 people at least, and they really rigorously assess it, and they will then come back to you. And with the paper we just published, they came back and we had to do 15 months of extra experiments to get it in that journal. And then often it might go through a second round. They might come back and ask for more experiments. So the peer review process is very important. It's very rigorous. But also importantly is to be going to conferences and really telling the world about what you found. So you might get a poster presentation, you might get an oral presentation, but very, very important to get out to the best conferences and be telling your colleagues about what you found. But also, even these days now, I'm even finding that social media, there's all of these new social media platforms, ResearchGate, LinkedIn, Yammer. There's all these different platforms where you can, get your research out there. And that's something I'm having to learn and catch up on. I have a bit of a phobia of social media. I don't like it, but the reality is, if you're not out there on social media, you fall behind. So it's published in Nature Communications, and it was published in June this year. That was 5 years worth of work, including the 15 extra months of after the peer review. Yeah, which is a very respectable journal. It actually was initially reviewed for a higher journal called Nature Medicine. It went to the 3 reviewers. We did everything they asked, but ultimately the editor still didn't like it, and they bumped it down to a slightly lower journal in the same publishing group called Nature Communications, but nevertheless still a very good journal. We've had good feedback. It's been reviewed by a few magazines. I've had quite a few emails from people. I mean, because it is only literally 2.5 months old, we'll see what happens, but based on the way I'd been advertising it while it was under review, I was invited to Madrid earlier this year. In fact, it was last September, and they paid for my flights, accommodation, registration to go and present that research at the premier HIV transmission meeting. So it's definitely exciting stuff. So my advice to students who want to pursue a career in scientific research is go in with your eyes open. It is a tough career. It's hard work. Doing a PhD is hard work. It's generally not particularly well paid, although you can, of course, go into pharmaceuticals or biotech and get good money. And also, for a long time, until you perhaps get a tenure position at university, it's not a very secure career, because you're dependent on grant funding. However, what I would say is, I think my personal view on life is that you have to follow your heart. And one of the things that I feel very sad about in life is just about how many people hate their jobs. It's almost just taken for granted that people hate going to work. It's just this horrible thing they don't like. I love my job. I never dread coming to work. And I would say one good thing about a career in science is it's fun. And you're working with people who are not valuing basically money and status as the be-all and end-all. You're working with people who want to lead a meaningful life, who want to make a difference, who want to advance human knowledge, and who are generally decent people. They're generally people who care, they're generally compassionate, they're generally kind, they're generally friendly. Of course, in any walk of life, you're going to find people that you like and you don't like. But I think as a lifestyle choice, science is really exciting, it's fun, and you really feel like you're making strides. When we first discovered that new cell, there was that real buzz of knowing that at least until your paper's published, you're the only person in the world who knows that cell exists, and that cell is the first one to pick up HIV and transmit it around the body. I think most scientists you meet are happy people. And I think as a lifestyle choice, it's certainly one to consider. It's never a boring life.